That is true. When you deacylate a histone, it helps open the nucleosome. So if they were to ask the opposite of this, that means if you acylate a histone, you are making it harder to open the nucleosome. Proteins recognize DNA due to their 3D shape complementary to the surface of DNA sequences. That's false. In transcription, we're looking for certain sequences like promoter sequences, things like that. So we're not looking for how well it binds to a shape of DNA, we're looking for specific sequences. If CTD is not phosphorylated, RNA polymerase II cannot start transcription. It's the other way around. CTD, when it's not phosphorylated, you can start transcription. So D is false. Which of the following statements is not true? Transcription is separated from translation in eukaryotes and prokaryotes. That's false. Yes?
What's CTD?
What is it? Cytosine something domain. I can't remember. I don't know if they tell you what the acronym means, but it is in your notes somewhere. And they mention that when it's not phosphorylated, transcription can begin. So in eukaryotes, transcription and translation are separated. Transcription occurs where in eukaryotes? In the nucleus, transcription in the nucleus, translation in the cytosol. In prokaryotes, though, is there a nucleus?
No.
No, so they are not separated. It is necessary for eukaryotic mRNA to go through post-transcriptional modifications. That is true. What type of post-transcriptional modifications happens with eukaryotic mRNA? Remove introns, GCAP, poly A tail. Only eukaryotic genes contain introns to be removed. Prokaryotes don't have introns. That is true. Because we don't do any post-transcriptional modification to prokaryotic mRNA, therefore it does not have introns to be removed. Guanylyl-transferase caps the pre-mRNA before leaving the nucleus for translation. We mentioned GCAP, that was one of the features. The 3' poly A tail of RNA indicates the termination of transcription. So it does indicate post-transcriptional modification. So you can only do post-transcriptional modification when you terminate transcription. So E is also true. This one cuts off.
Josh, help us. Where are you going to send these?
Right away. Yeah, I have to change a few of these because there are some mistakes. Yeah, you want me to highlight the answers or put them on a separate sheet?
Just highlight them, please.
Highlight.
Okay. Yeah. All right, more true and false. Actually, did we do this yet? Yeah, actually, this is the last one. In structure motifs, most contacts occur in the major groove of the DNA.
True.
That is true. Remember, major groove is where a lot of regulation occurs. The minor groove does not have that. There are two main classes of the zinc fingers. What are they? C2H2 and CX are the two zinc finger classes. In zinc fingers, only a folded beta strand fits into the DNA major groove. That is false. Both types can fit into the major groove. Heterogeneous nuclear RNA, also known as hnRNA, is exported to the cytosol. And let's say we're talking about eukaryotes. Is that true? That's false. Remember that hnRNA is another way of saying pre-mRNA. And we know in eukaryotes you must first edit it. You must add a GCAP, poly A tail, and remove introns. And the last one, chromatin is part of the complexity of eukaryotic transcription. Chromatin, with all the histones, with all the nucleosomes, makes transcription a little bit more regulated in eukaryotes, so that is true as well. Okay? All right on time. If you have any other questions, let me know. I will send them to you after I finish a few things, but I'll have to answer